Protein synthesis is essential to our body because it helps your body transport messages, supports the structure of the membrane, and it also protects your body. So, in order to get the wanted protein from the nucleus to the ribosome in the cytoplasm, a copy of a selection of DNA must be made to send messages to the ribosome. This process of copying the selection is called transcription. It happens in the nucleus with the help of enzyme named RNA polymerase. It unzips DNA and builds new strands of nucleotide that concludes in a new strand of RNA called mRNA or messenger RNA. This process happens in the leading strand of the DNA. So remember, it only takes place in part of DNA. Here is the complicated part of the process. You may remember that in a lot of processes, adenine matches with thymine and cytosine matches up with guanine. But anytime the mRNA places a complement to an adenine on the DNA, it places uracils. This is because RNA uses uracils instead of thymine. After clipping on the complement of nucleotide on the DNA zipped back up and mRNA started to go to the ribosome and transfer messages. Some part of the RNA are left behind, such as introns. Introns are gene sequences that are not expressed in the protein, so it stays behind. On the other hand, exons can go out of the nucleus from the nuclear pore and express protein. So let me go on to the translation part. Then the process moves on to the translation, which happens in the cytoplasm. When the mRNA is attached to the ribosome, the codons were usually started with AUG, then the tRNA connects to the complement match of the mRNA. In between the step of connecting the tRNA and the mRNA, there is RNA. The RNA is the protein making machine that reads the mRNA and put the correct amino acid using the tRNA. The tRNA has anticodon and mRNA contains codons in which they connect each other. Remember, codons are 3 nucleotides and tRNA reads the base of the nucleotide in 3. When the other tRNA moves in and matches up with the mRNA codon, the amino acid form peptide bonds. And this process goes on until it reaches the stop codon. When the process is done, the proteins are formed and are ready to be used. To know the sequence of the amino acid, we can use the mRNA and the amino acid chart. This is the chart of the amino acid and the mRNA. Okay.